With every trial, every test, we move closer to the rest. A world where pain no longer stays illuminated by brighter days. Waits for a cure. Rise and shine, rise and shine.
Okay, good morning, everyone. Sorry for the last minute thing. My name is Nicolas Chomont from University of Montreal. I'm the director of the Canadian Intervention Enterprise. And first, I would like to thank the organizer of this community workshop for the invitation. It's really a pleasure to be here. Not my first time attending this community workshop just before CROI. And again, very happy to present CanCure today. So before I go into the details of CanCure, I'd like to give you a picture of the HIV epidemic in Canada. And actually, the numbers are not good. They are not good for several reasons that I will not detail. I'm sorry? Oops. Yeah. So let me see. I'm not sure I can do that.
Close. I guess we can't get it.
I don't know why we can't.
Well,
Michael?
Well, it's not on mine.
Okay. You just reshare. Sometimes you can reshare.
Okay.
we'll stop the sharing and then we'll share it again. Oh, that's because we stopped sharing.
Yeah.
So let's see.
This meeting is being okay. Right. I'll try again. So we should. That's probably because of this. Okay.
It's just letting you know.
Is this the one?
Yeah.
Okay. Can you see that online?
Yes.
Okay. So we're good. All good. Wonderful. So the HIV epidemic in Canada, we are not very good. We are not very good in the sense that the incidence of HIV is increasing in Canada, particularly in the prairies. So in the Saskatchewan and Manitoba, which are the two provinces that you can see in brown on this picture. There was a 35% increase in the HIV incidence between 2022 and 2023 for multiple reasons. But the reason I'm showing this slide is because, sometimes people ask me, why are you still working on the development of a cure for HIV? Well, this is not only a Canadian thing. If you look at the numbers in the world, because of the funding restriction, the recent funding restriction from the US administration this year, there is clearly a projected increase in new cases of HIV and probably in HIV-related deaths that will occur in the next 5 to 10 years. And these restrictions could potentially put us back to levels that we have not seen since the early 2000s. So I would say that we cannot probably depend only on the support of the US government, obviously, and not only on antiretroviral therapy to contain the HIV epidemic, and the cure is probably more needed than ever. A part of this, the life expectancy of people living with HIV gets better, and this is shown on this slide, and you can see the reduced gap between people with HIV and people without HIV. But if you look at the comorbidities, we still have a lot of work to do. Basically, people with HIV develop comorbidities on average 15 years earlier than people who do not live with HIV. So there is a higher and stable occurrence rate of comorbidities in people with HIV that is not, or that are not normalized by antiretroviral therapy. So we need to do better than this. Some people might still ask, why do we need a cure? This is a slide I usually present to audiences that are not very familiar with HIV or with HIV cure. But I think you guys in this room know that very well. But it's always important to remind that antiretroviral therapy is not ideal for many reasons. First, it does not resolve the issue of stigmatization and criminalization of people living with HIV. Many people, and more than ever this year, do not have access to antiretroviral therapy. There is some toxicity associated with the treatment or with the drugs. There is obviously an economic burden of HIV, and there is, more importantly, persistent inflammation and higher rates of comorbidities in people living with HIV compared to the general population. So we clearly need to do better, and to do better, we need to eradicate the virus entirely. So here's the next question: why we don't have a cure for HIV? So as you know, when people start antiretroviral therapy, very rapidly the viral load, so the amount of virus circulating in the blood, is decreasing, and these levels will actually reach undetectable levels by clinical assays in a few weeks. Unfortunately, when people stop taking antiretroviral therapy, the virus comes back quite rapidly in a matter of weeks. So it's pretty clear to us now that antiretroviral therapy does not eradicate the virus. And what we are trying to do is to understand what's happening during this phase when we don't see the virus in the plasma, but obviously it's still hiding somewhere. And what we are trying to do, like many other people around the world, is to identify what we call these reservoirs and develop strategies to eradicate this virus. And our goal is to develop a cure, and the way we define cure, and there are several definitions of a cure, obviously, is that we want people to be able to live without antiretroviral therapy, without virus, without comorbidities, and without any likelihood of transmitting the virus to someone else. So what is CanCure? CanCure is a multidisciplinary team with a common objective. We want to translate basic knowledge on HIV reservoirs and persistence into therapeutic approaches. And our ultimate goal is to develop a safe and scalable cure for HIV infection. And we do that by identifying biological mechanisms that contribute to the persistence of HIV after prolonged therapy, and that can be therapeutically targeted to eliminate the reservoir. Our goal, again, is to control viral resurgence upon ART interruption. The CanCure team includes immunologists, virologists, clinical researchers, and more importantly, community representatives. And Shari Margolis will say a word about our community advisory board, CanCure CAB, which is composed of people living with HIV and other people representing other communities affected by HIV. So this is the third edition of CanCure. It's funded by the CIHR. CIHR is the Canadian NIH, if you would. And CanCure is funded through a competitive program, which is called the HIV and STBBIs. STBBIs is for sexually transmitted blood-borne infection, a term we are using quite widely in Canada. This is the third edition of CanCure again. CanCure 1.0 and 2.0 were led by Eric Cohen from IRCM in Montreal, and I have the honor of directing the third edition of CanCure. The period of performance of CanCure 3.0 is January 2025, so we started a little bit more than a year ago until December 2029, and our budget is $3.75 million over 5 years, which is probably 5 times or 10 times less than the large NIH-funded consortium. So we're doing with less money, I would say, but I think we are doing good still, and I'll let you decide on this. This is the CanCure team. So again, a collection of people from all different places in Canada, from British Columbia on your left I was about to say Mexico, but Mexico has been moved on this slide, obviously, but to Prince Edward, Nova Scotia, thank you, and even Newfoundland and Labrador. I can use actually my arrow here, I think. So again, scientists, yes, clinician, basic scientists, but also our community advisory board, which is led by Shai Margolis that you can see here on this picture. And we have collaborators in Mexico listed on this slide. Okay, the CanCure agenda is really focusing on the tissues. And the reason we're doing this is because we know, and several studies, sorry.
I can hold this in.
Oh, I'm good, thank you. The reason for this is because we know that most of the HIV reservoir cells hide in tissues. So I will not detail all the things that we are doing into CanCure. I'm going to give you some examples of the work we're doing. What I think is important to remember is that we have a first theme, which is really on the identification of these reservoir cells in tissues. Another team which is really about the immune response. So how can we manipulate the immune response to better control HIV replication? And our third theme is really about therapeutic interventions. We are doing this in animal models, but also in humans, and CanCure has several clinical pilot studies embedded within its program. And to do all this, we have access, obviously, to tissues from participants, from people living with HIV. I will mention also our end-of-life program. And of course, we also use animal models. Okay, so you probably know these. There are multiple ways we can try to eradicate or control the virus. The first one is to limit the establishment of the reservoir by giving ART antiretroviral therapy. Obviously, many people are late diagnosed, so this is not something that would be very realistic. So we need other strategies to target and reduce this viral reservoir, which is the cause of viral rebound when ART is interrupted. And there are several of these strategies listed here. Also, another part is to boost the natural immune response of people living with HIV to try to naturally control the virus. And CanCure is actually involved in several of these strategies, which are indicated here by the maple leaf. So again, I will briefly describe the objectives and themes of CanCure. You can have access to these slides. If you want me to, I can share them with you. We have three teams. So the first one, mainly virology, the identification of the reservoirs. The second theme is more about immunology, boosting the immune response to control the virus. And the third theme is to develop new interventions in animal models and in humans. So again, team one, it's really about identifying the reservoir cells. And with this particular focus on tissues, I will detail in a second what we are doing with this end-of-life study, which is the equivalent of the last GIFT study from UCSD. In team two, more immunology, but again, with a big focus on tissues. And as you can see, we have many investigators involved in these studies listed on this slide. We are also very much interested in the metabolism of HIV reservoir cells, but most importantly, on the metabolism of HIV immune cells and how we can make them better fighters against the virus. And finally, in this third team, we are doing three different things right now. We are trying to target CCR5, pretty much like to replicate what has been done in these cases of people who have been cured after stem cell transplant, but making something more accessible, I would say. We're using oncolytic viruses. For the sake of time, I won't detail the studies. And we also have this fecal microbiota transplantation program, and I'll be happy to answer any question you may have on this. The tissue sources are multiple from the Canadian end-of-life study, which is led by Cecilia Kosteniuk, with sampling in people living with HIV, both in Canada. This is a part led by Jean-Pierre Routy in Montreal, but also from our collaborators in Mexico with Santiago Avila and Gonzalo Salgado. We also have a program to access lung cells by bronchoalveolar lavage. We also have a program to access non-human primate tissues, and finally, a humanized mice model. This is a boring slide with the leadership of CanCure. I'm sorry for this. I need to mention it. So obviously, we have the steering committee, and I have to mention that Shari, who is here with our principal knowledge user and the director of the CAB, is part of the steering committee. We also have a scientific advisory board. Okay, so I'll take the last part of my talk to really detail what we're doing in CanCure. So our approach, again, is on tissue reservoirs, trying to identify where the virus hides. Then we try to develop approaches to better characterize at the cellular level within these tissues what are the reservoir cells. And finally, we try to target therapeutically this reservoir cell. So our hope is to develop a cure by coordinating these three aspects of our research. So let's start with tissue reservoir. Where does HIV hide? This is, of course, a very important question. Again, a question that has been addressed by multiple studies during the past 20 years. And really, the pioneers in this field are the last gift study from UCSD, led by Sarah Gianella, Antoine Chaillon, Karine Dubé, and also many people in this room, including Jeff Teller and Michael and others. So thank you very much for leading this effort, which was very inspiring to the Canadian scientists. We've learned from the last gift study that HIV persists in many deep tissues. And in Canada, we have a similar study again, which we call the end-of-life study. We published data on 2 different participants. This is a geographical test to see if you're good in Canadian geography. Do you know where is Ottawa and where is Edmonton? Can you guess? Yeah, so here's the answer. So Ottawa is on the right, Edmonton is on the left, and we recruited the first 2 participants from these 2 cities. They were both in their 60s. The first one from the participant from Ottawa had medical assisted death, and the second one died or passed away from a B-cell lymphoma. They were both diagnosed during the 80s, and they generously gave their body for research on HIV reservoirs. So through a rapid autopsy program, we were able to collect many deep tissues from these 2 participants. And this is the collection of tissues we were actually able to collect. With, of course, a particular focus on what we call the lymphoid tissues. So lymphoid tissues are tissues of the immune system. These are your lymph nodes, your spleen, and the reason why, and also your gut. And the reason why we're focusing on this is because previous studies have shown that they are enriched in the HIV reservoir. I'm not going to spend too much time on the detail of these studies, which are already published, but the basic finding is that we found traces of HIV pretty much in every single tissue we looked at. This was true, of course, in the lymph node, in the spleen, in the liver, in the lungs, and also to some extent in the brain and in the testes. What we do within CanCure is that we try to push the limits of the technology to better characterize the reservoirs, and we use more sophisticated assay to detect what we call the real reservoir. So the distinction between what we showed in this previous slide and what we have done here is to actually be more precise in the analysis we're using to really detect the real virus, because most of these viruses are actually not able to replicate in culture. So it's important to differentiate between the real reservoir, the one that can cause rebounds when art is interrupted, from, I would say, the graveyard reservoir, which is made of dead viruses. And using sophisticated approach, we can do this. And so when we did this, we realized that actually most of the genomes or the HIV that we found in these people were defective, not a real reservoir. And it's only a very tiny fraction of the reservoir that you can see here. 1.7% of all the cells in which we find traces of HIV are actually a real reservoir. So the background, or the noise, I would say, is huge, and we really need to perform this sophisticated analysis if we want to have a clinically meaningful answer, I would say. And when we focus on these intact proviruses, so the real ones, it's really clear that they were concentrated in the spleen and in the lymph nodes, so again, in the lymphoid tissues. The picture was a little bit different in the other participants. We have several hypotheses to explain this. I can go back if you have questions on this. Okay, I'm done, almost done. This was for the tissue reservoir. For the single cell reservoir, again, this is work that has been done in my lab by Hélène Roux, who was a postdoc, who's actually now back in France. And Hélène used samples from 11 people living with HIV. Unfortunately, all males. These people were enrolled in Montreal between 35 and 60 years old. And we sampled multiple time points from them. And we did very detailed analysis of the reservoir in these participants. I'm not going to detail what we're doing here, but this technology that we developed in our lab with the help of many other partners within CanCure really allows us to characterize the reservoir, what we call at the single cell level. So a very detailed analysis of the reservoir. And on this slide, what you're seeing in red are HIV reservoir cells, the red cells, and what you're seeing in gray are the non-infected cells, so the majority of the cells in the body. And even if you don't understand flow cytometry, I think you can see right away that most of the uninfected cells in gray do not cluster with the red cells, which are the HIV-infected cells in red. And so this means that these HIV reservoir cells have really particular features. They are very, I would say, weird, actually, very weird from an immunological point of view. And the fact that they are weird gives us an opportunity to target them. So we did that in multiple participants, multiple time points. And again, we find when we compare the phenotype or the face or the ID of these reservoir cells, which is here, compared to the non-infected cells, that they express several genes at higher levels, like granzyme E, interferon gamma, TBET, and KRG1. And so we're trying to better understand what this means. But the good news is that this means that this HIV reservoir cell, which is a very tiny portion of all cells in the body, have really special characteristics that might be targeted. So we try to understand what does that mean? Why these cells are so different? And we think they are very different than the others because they have this exquisite capacity to persist for many, many years. Of course, if you acquired HIV 20 years ago and started antiretroviral therapy right away or a few years later, it means that you basically froze the system. So all the cells that got infected at when you acquired HIV had this capacity to survive for decades. And this capacity to survive is actually something very peculiar, I would say, that would provide a signature of these HIV reservoir cells. And we believe that some of these cells that persist for many, many years actually are what we call in immunology senescent T cells. And so based on this knowledge, then we're trying to therapeutically target these cells. And this is something that other people have done before us, particularly here in the US with Adam Spivak, but also in Spain with Maite Carreras, Beatriz Mote, and Pepe Alcami. And the good news is that there are drugs approved that can specifically destroy these senescent T cells. And since many of these HIV reservoir cells display this senescent phenotype, we believe that we could actually get rid of some of the reservoir by using these drugs. So this is probably too early to actually do that in people, but we are doing this in the lab from now. And we tested two different drugs, which are called quercetin and fisetin. And as you can see, if you compare without treatment or with treatment, this is the number of reservoir cells in a well in the lab. We were able to reduce significantly the number of reservoir cells. So very preliminary data, but really encouraging. So I think this is a nice demonstration of what we do within CanCure. We start from the tissue, then to the cell, then we try to implement or develop therapeutic strategies based on this. And of course, our next step is to do that in clinical models and hopefully in humans. So this is my summary for everyone. So CanCure is a Canadian consortium for scientists, clinicians, and community representatives working on a common objective. We look at the virus in its natural environment, which are tissues, to better understand the way it persists. As you've seen, it persists in many tissues, but the lymphoid tissues seems to be very enriched in the real reservoir. So I think these are the tissues we should primarily target. We use sophisticated approach to identify the characteristics of these rare reservoir cells. And many of these reservoir cells are old cells, and our in vitro data suggest that they can be depleted using some drugs called senolytics. And even though this is still very preliminary and needs to be validated, we're actually quite excited by these findings. This is part of the CanCure team. I'd like to thank all the members of CanCure who have worked incredibly hard for the past 10 years on the different scientific priorities of CanCure. And finally, I'd like to thank some members of my lab. I'd like to thank the CanCure CAB. The members of CanCure CAB are listed here. The funders, so mainly CanCure and CIHR. We still have a very good relationship with the NIH, and I'm part also actually of DARE, which is one of the NIH consortium funded. So you might see me as promiscuous because I'm both with CanCure and DARE. But actually, this is really cross-fertilizing, and I'm really grateful to my American colleagues for still supporting me in what we're doing in Canada. And finally, I obviously need to thank the study participants to these studies. I mean, obviously, all the work we're doing within CanCure is because we have very committed participants who are giving samples, their time, and without them, nothing would be possible. Thank you very much.
Thank you so much. Oh, yes.
You still have a mic.
So there are some questions in the chat. Another round of applause for Nicolas for not only keeping us on time, but doing an amazing job. Thank you so much. So one of the questions in the chat, and Richard actually addressed it in the chat, but I want to give you an opportunity to answer it. Jill says, let me go back to it. Can you please explain the difference between a real reservoir cell and not a real one?
Yeah, thank you for the question. So, yes, that's a very good question and something that has been discovered actually less than 10 years. It's a really recent discovery coming from Silicano and many others. So basically, HIV is a virus that is really bad at replicating itself. And when it does, it introduces a lot of errors in its genome. The reason it's doing this because by introducing errors randomly, it can gives it a selective advantage to actually evolve in a good way. But most often, I would say, it creates mutations that so it kills the virus, but the virus is still archived in some of the cells. So many of the virus that you can find in cells from people who have been in art for many years still have these traces of errors, things that would not allow the virus to replicate. So when we measure the reservoir, we should ignore these dead viruses and really focus on the real ones, which represent again less than 5% of all the
Sorry to interrupt, the people online can't hear. We might be
Sorry.
the laptop.
Okay, I'm not going to repeat all this, but so basically the difference between the real and the not real is that the real one has the ability to produce new viral particles, new virions, new viruses, and can reignite infection, while the dead viruses can't. And so it's very important to distinguish the two. And using sophisticated assays, this is something that is feasible.
I have a question in the audience. I'm going to grab that. I'll be right back.
Excellent presentation. Thank you again. My question is, and it's an observation that I have, but I learn things based on my questions that I ask. Has there been any findings, indicators in reference to, I did not see any women in the studies, but women have a reproductive system, and not all of us who are living with the virus, get hysterectomies, and so we still have, within, those origins within our bodies. Do you know if there's anything related, and I'm asking this because of what menopause does to many of us as we're aging. Do you know if there has been any identities of reservoirs that comes from a woman's reproductive system, looked into?
Yeah.
Could briefly restate the question.
Sure. So the question is whether, briefly, whether the reservoir in men and women differs and whether the reproductive tract can contribute to HIV reservoirs as well. That's a very good question. So there are not so many work, more and more actually, showing that yes, there are cells in the reproductive tract, both in men and women actually, that can host the virus. So these organs can be HIV reservoir. More importantly, there are fundamental differences between men and women in the way these cells persist. Men and women are not the same biologically, and hormones probably play a role in that sense. Aileen Scully has done a lot of work on this to demonstrate that the size of the reservoir is not the same in men and women. And also the inflammation is probably much higher, is a slightly higher in men than in women, which contributes to which contributes, I would say, differentially to the persistence of the reservoir in men and women. So yes, it's very important to actually distinguish the results findings from these studies between men and women. I'm ashamed to say that in the study I just showed you, 11 men were enrolled, not a single woman. It's not good, and I apologize for this.
Guys hear me on this? I'm just gonna use my outside voice. Thank you for that. Are there other questions in the room? Ms. Campbell, can I use your outside voice?
Okay, so thank you for following up on your question with regards to the lack of representation of women in studies. What's your plan of amelioration?
Yeah.
I appreciate the sincere,
So we have multiple plans because this is something that we're trying to address very seriously. So of course, the easy answer, when you write a grant and when you say what's going to be your population, and we always say, well, the enrollment in your studies reflect the populations of people living with HIV in Montreal, but that's not enough. Because even though it's true that I don't have actually the real numbers, but in Montreal only more than 70% of the people living with HIV are men. Still, that does not explain why we don't have women in our studies. So the clinicians we're working with, particularly Jean-Pierre Routy in Montreal, has now developed several approaches to enforce, that's probably not the right word, but favor the recruitment of women. So that means actually saying no to some of the men who would like to enroll in some studies to maintain the equilibrium. So that's one way of doing this, which is not ideal. There are other sites in Canada that are much better than we are, and particularly in British Columbia in the recruitment of women. So we should probably get inspired by what they are doing to do this. But we still have a lot of progress to make.
Do you have any other questions in the chat?
yeah, there's one question. What's the reason behind the wide gap and the real reservoir percentages among different patients?
Sorry, say that again.
What's the reason behind the wide gap in the real reservoir percentages among the different patients?
Yeah, that's a good question. So some people.
Frustrating.
Sorry, yeah. So the real reservoir, the size of the real reservoir can differ between people. So the reason for this, we're not exactly sure, and particularly in the two cases I showed you. So we have two participants. In the participant from Ottawa, the real reservoir was only 2%. In the participant from Edmonton, it was 25%. We believe that comorbidities probably play a role in this. I did not detail all the comorbidities that the participant from Edmonton was living with, but inflammation can maybe induce residual replication, which could explain why the real reservoir was much higher in this participant. So that's one possible explanation. I'm sure there are so many others.
And then I had a question, if you don't mind.
Yeah.
So you mentioned that you're big collaborating with some US sites, and of course, with the past year, you're not allowed to send any money to outside investigators. How has that affected your research?
Right. Thank you for the question. So basically, honestly, what happened to me on May 1st, 2025, I lost abruptly 50% of the budget of my lab. Because 50% of my activities were funded by NIH grants. So don't get me wrong. First, I am very grateful to the NIH and the American people for funding my lab for many, many years. And I will always be grateful for this. The problem of the abrupt funding that we experienced last year is that this money was paying the salary of people who have been working with me for 10, 20 years. The salary or the fellowship of students who were in the middle of their PhD, basically. So that was a dramatic situation to deal with. These funds have not been reinstated to the foreign country or to other countries. So, but I'm grateful to my university who actually helped me a lot and said, well, you've been funded by the NIH for so many years. Congratulations. Now it's a difficult time for you. So we will support you by giving you money to support the salary and the fellowship of the students. And thanks to them, I did not have to fire anyone in my team. So that was really a relief. Now, I don't know what the future is going to be made of, who knows? I have to say that this is something that we will address at CROI in the next 3 days for those who will be attending the conference. Of course, the cut in funding for research is dramatic. The USAID cut are even more dramatic because we're talking about people's lives. I'm not an American citizen, so I'm not going to tell you what you should do. The best we can do is to gather like we do today, to show our solidarity and to resist.
Great, we have one more question. We have time for one more question, and I'm going to take it out of the chat. Okay. Patricia asks, how receptive are people to cure approaches, and what work is being done with community participatory research to address concerns and challenges of First Nation communities?
Wow, that's an excellent question, probably more for Shari, and Shari will address this in a much better way than I will. This is
Through the mic, if you want to.
Oh yeah, sure, Shari, you'd like to address this?
Oh, actually, you have to clear that so they can hear in the chat. Very great. She's coming, guys. Stand by.
I can put you.
So for the work that we're doing, two of our CAB members, and we have a very small community advisory board, are First Nations women. And they are very well connected to the system that we have that in the community of different organizations working with First Nation, Métis and Inuit people in Canada. We also have collaborations with the national Indigenous group called CAN. And we also are currently, as a matter of fact, right now, we're going to Winnipeg for our national conference in April. And there's a very large First Nations and Métis population there. And we are partnering with some of our local people there that I've worked with over the years in many other projects and things to talk about, and we're bringing in an elder for that session as well. You don't know that yet, Nicolas, that just got confirmed. And we'll be having a whole session grounded with an opening and closing from the elder. We'll have a panel to talk about what does a cure mean to them? What do they think about all this research? We've also done some studies, but I'm going to talk a little bit about that as we go through my piece. So yeah, we are grounded there. And in Canada, this is a really big deal. We really value our First Nations, Métis and Inuit communities and try to include them as partners. And also, a lot of our research teams doing different kinds of research across the country are, talking about governance of data and those kinds of things where they actually hold their own data. So it's a kind of an evolving area because more and more now, when I first started doing this, I worked with like in the 90s, worked with a provincial or regional group, and we developed a Quay to Quay called Woman to Woman program, because really what we were trying to do was to get more people engaged. Because for many years, there were only a dozen of us who did media work, who were public, who were really engaged in research. And now we have so many. So we can talk about that more offline too.
Quest for a cure.